Remember, the optional final exam is optional. You do not have to take it, but if you do take it, if the grade is higher than your lowest lecture exam grade, it will replace that one. If your optional exam grade is the lowest grade, it does not count. And we will have both an optional lecture final and an optional lab final. They are both cumulative. And for you
Wait,
guys,
Professor,
yes.
you said
Uh-huh.
optional lab final. Is that
Yep.
what you're doing today?
No, today is lab exam 2, but then tomorrow there'll be an optional lab exam as well.
Oh, so we have optional lab exam tomorrow and the optional lecture exam tomorrow.
Yep, and you have all day until
So are
11:59
they going
p.m.
to be like together or we do them
Oh
together?
no, they're separate, so I take whichever one you want, or both. Any other questions? So we started the course by briefly talking a little bit about evolution. Not that much, but a little bit. We do know that Charles Darwin came up with this theory of natural selection and the idea that those members of a population that have the most beneficial characteristics will survive better, reproduce more, their offspring will survive better, and eventually the species evolves. And so there's always natural variation within a species. That natural variation is mostly the result of random chance mutations. Some of those mutations are beneficial, most of them are not, right? They're either harmful or neutral mutations, but the accumulation of beneficial mutations leads to evolution. And so Darwin made observations on the Galapagos Island, in particular with the species of finches that were on that island. So there were a bunch of different finches on the island, but they all descended from one ancestral species. And over a long period of time, many different species evolved in order to exploit the different resources on the Galapagos. Because there's not enough resources, so if they all ate the exact same thing in the exact same way, then most likely the finches would have died out, or you would have been left with only one or two species. But we have a bunch of different species, and that's because they evolved different characteristics. If you look at the shape of their beaks, right, their bills, you notice that they're different because they're specialized, for eating insects or seeds. And so we have all these different species which came from one original ancestral species. And over time, mutations developed and it allowed the different species to evolve. Okay, wait a minute. My thing was stuck. Okay. So we talked a lot about energy. Remember that energy is the capacity to cause change. And there are several different types of energy: kinetic energy, right, the energy of motion, potential energy, thermal energy, chemical energy. And enthalpy is a measure of the total energy of a thermodynamic system. So all the energy that exists in that system and whatever energy was needed to create that system. So it's all the intrinsic energy of that system. Entropy is a measure of disorder or randomness of a system. Usually the universe, and we know the entropy of the universe is always increasing, although the entropy for a particular system may decrease. We use the example of, a complex multicellular organism like us. We are very highly ordered systems. And the reason why we're ordered systems is because we have to input a lot of energy to maintain that order. If we stop inputting that energy, then our system will become very disordered because we die and begin to decompose. So we become much more random. The first law of thermodynamics states that the energy of the universe is constant. So energy is neither created nor destroyed. It's simply transformed from one type of energy to another, or it's transferred from one system to another. So an example that I always use was an old-fashioned light bulb, where the light bulb produces light energy and heat energy, right? Because it gets hot. But the amount of energy from the light and the heat is equal to the amount of electrical energy that is producing that light and heat. The electrical energy is merely being transformed from one type of energy to two types of energy, the heat and light. So the energy of the universe is constant. The second law of thermodynamics states that every energy transfer or transformation increases the entropy of the universe. So we know with all different chemical reactions, some energy is lost to the environment, usually as heat. So that's heat energy being released, to the environment. So it's being released back to the universe to increase the entropy of the universe. When it comes to reactions, an exothermic reaction is a reaction that releases heat energy. So the temperature increases with an exothermic reaction. An endothermic reaction is the opposite. It absorbs heat energy from the environment, so the temperature decreases. Exergonic reactions are those reactions where the enthalpy or the energy of the reactants is greater than the products. So the reaction proceeds spontaneously. So in other words, the products are lower in energy, so they're more stable. So that's why the reaction proceeds spontaneously, because it's going from less stable to more stable. And energy will be released when that happens. The delta G, right, the Gibbs free energy is negative, and the entropy increases. So again, energy will be released. An example of an exergonic reaction is aerobic cell respiration. An endergonic reaction is a reaction where the enthalpy or the energy of the products is greater than the reactants. So the products are less stable than the reactants. That's not a favorable reaction. It's not going to occur spontaneously. So in order to make that reaction happen, energy must be input into the system in order to make that reaction occur. So the delta G is positive. The entropy of the system, not the universe, usually decreases. So we talk about forming chemical bonds or building molecules. So anabolic reactions are endergonic. And remember, we said the primary way that cells make endergonic reactions occur spontaneously is to couple those reactions with the hydrolysis of ATP. ATP has a very negative delta G. So the hydrolysis of ATP into ADP is spontaneous. You can couple that reaction to an endergonic reaction. The overall delta G becomes negative, so the entire reaction will become spontaneous. That's the main reason why cells need ATP, is to make these necessary endergonic reactions occur spontaneously. So we talked about different cell types. We have eukaryotes versus prokaryotes. Remember, eukaryotes, the main difference is they have a nucleus and membrane-bound organelles like mitochondria, chloroplasts, Golgi, endoplasmic reticulum. You do not find any of those organelles in prokaryotes. No nucleus, no membrane-bound organelles. So prokaryotes are much simpler cells. Animal cells and plant cells have a lot in common. The two major differences is that animal cells just have a membrane that surrounds them, and plant cells have a membrane plus a cell wall made out of cellulose. Plant cells also have chloroplasts, so they can perform photosynthesis. So the organelles, I mentioned most of them, the energy-producing organelles, mitochondria and chloroplasts. The nucleus, where most of the DNA is stored, not all of it. Remember, mitochondria and chloroplasts had a small circular chromosome. And we talked about how mitochondria and chloroplasts are believed to have been derived from or evolved from prokaryotic cells. We call that the endosymbiotic theory, where one cell swallowed another prokaryotic cell, but instead of eating it, they lived together in harmony. And over many millennia or eons, the symbiont, the cell that got swallowed, specialized for energy production because it did not need to perform most of the other cellular functions because it was living inside of a cell. And the host cell was doing everything else needed for a cell to survive. Talked about the endoplasmic reticulum, which remember is touching the nuclear membrane, and ribosomes are attached to the endoplasmic reticulum. Sometimes we call that the rough ER. There's other endoplasmic reticulum, which is the smooth ER. But for the rough ER that has the ribosomes attached, newly synthesized polypeptides move into the endoplasmic reticulum, where they may be modified by having sugars added onto them to make a glycoprotein, or fats added onto them to make a lipoprotein. But they are packaged inside of the endoplasmic reticulum for delivery to the Golgi, and from the Golgi to wherever they're needed inside of a cell. If they're secreted, a vesicle buds from the Golgi, heads to the plasma membrane to fuse with the plasma membrane, so the contents can be released. Otherwise, the proteins can be delivered wherever they need to go inside of the cell. Because the proteins have information attached to them, like a zip code, a postal code, which will tell the cell where to deliver, right, those proteins. So here we see some examples. Our animal cell, nucleus, lots of endoplasmic reticulum, mitochondria, just a cell membrane. There may be structures like flagella which help in movement. A plant cell, mostly the same, except there's a cell wall, chloroplasts, and there may be a storage vacuole. Like in potato cells, they store starch inside of a vacuole. And prokaryotes have none of that stuff. Right? They're much simpler cells. They have a cell membrane. Most prokaryotes have some type of cell wall. They may have flagella on them, which enable them to swim. And they may have other sticky structures attached to them to help them stick to other cells or stick to substrates. But no nucleus, no mitochondria, no organelles. So we did some basic chemistry. We learned that an element's atomic number is the number of protons in the nucleus. Remember, protons have a positive charge. Their charge is balanced by having the same number of electrons, which are negatively charged. The mass number for an element is the number of protons plus neutrons. Remember, neutrons have a neutral charge, but protons plus neutrons is the mass number. When you look at the atomic symbol, it's not all done the exact same way, but the larger number that's displayed is the mass number, the number of protons and neutrons, and the smaller number is just the number of protons. Now here we have carbon-12, right? The most common form of carbon. But if you had something like carbon-14, that would be an isotope of carbon. And carbon-14 would contain 2 extra neutrons. So isotopes of an element are the same except that they contain extra neutrons or different numbers of neutrons. We talked a lot about chemical bonds and how important chemical bonds were when it comes to molecular shape and function. So some bonds are polar bonds. So a covalent bond is one in which two or more electrons are shared between two atoms. If the atoms are basically equal in terms of their affinity for those electrons, and the fancy word is electronegativity, if they're pretty much the same or equal, then the electrons are shared equally, and that bond is a non-polar covalent bond. If, however, you have an atom that's much bigger, more powerful, and here we're looking at H2O, so the oxygen is much bigger, much more powerful than those little hydrogens, then the electrons are pulled closer to the oxygen. That gives the oxygen a partial negative charge and leaves the hydrogens with a partial positive charge. They are still sharing electrons between those atoms, so it's still a covalent bond, but the electrons are not being shared equally, right? They're closer to the oxygen than they are to the hydrogen, so that creates a polar covalent bond. And because of the special property of all these partial charges, the partial positives on the hydrogens and partial negative charge on the oxygen, that allows for hydrogen bonds to form. So hydrogen bonds occur between the hydrogens of molecules like water and the oxygens of another molecule of water, or they can happen between molecules of ammonia, NH3, where the same situation occurs. The ammonia is big and powerful and pulls those electrons closer towards it, giving it a partial negative charge, and all the hydrogens have a partial positive charge. And so the hydrogens can form a hydrogen bond with a slight negative charge of a larger atom like oxygen or nitrogen. Those hydrogen bonds give water some very special properties, which we'll get to next. But don't forget about ionic bonds. So ionic bonds are full charges on those atoms. And the most common example we use is NaCl, where the sodium is a positive charge and the chlorine has a negative charge. And that's because the sodium had an unpaired electron in its outermost shell. And we know that electrons do not like to be alone, right? They like to be together in pairs. And so sodium has this extra electron in its outermost shell. And the chlorine, it is missing an electron in its outermost shell. So the sodium donates that electron to the chlorine. And when that happens, because the sodium is losing a negative electron, it leaves a net positive charge on the sodium. And the chlorine, it's gaining that negative electron. So it now has a net negative charge. And so we call these ions now. So sodium is a cation because it's positively charged, and the chloride ion now is an anion because it's negatively charged. And so they can interact with each other in an aqueous solution, in a watery solution. And those water molecules will surround the sodium and chloride ions because water has partial charges on the hydrogens and the oxygens, so they can surround, right, the sodium and the chlorine. And that allows for the sodium chloride to dissolve in water. Now back to why is water sticky? It's all those hydrogen bonds. It makes water sticky, right? Because the water molecules can interact with each other and form these hydrogen bonds. And there are two very special properties of this stickiness that occur: cohesion and adhesion. Cohesion is where the water molecules stick to each other and create surface tension. So if you've ever had a nice washed wax car and then it rains, on the car we see that the water beads, it stands up. It doesn't spread out, it stands up. And that's because of cohesion, the water sticking to itself. There are some insects which can walk on water because they do not break the surface tension. They don't fall through, so they're actually able to walk on water. And that's due to cohesion. Adhesion is where the water molecules stick to other things, like the xylem in a plant, the little tubes inside a plant, where the water gets absorbed through the roots, moves up through the stem, and evaporates to the leaves. Plants don't have hearts, right? They don't have pumps, but the water moves up because of something called capillary action. So the water molecules grab onto the sides of the xylem, the little tubes in the plant, and then they pull more water molecules up behind them. And then they grab on and stick to the tubes and pull more water molecules up. And so water moves up through the xylem of a plant. Humans have these really small, thin glass tubes called capillary tubes. And what they do is they can collect, they prick you and you get a little drop of blood and you put a capillary tube into the blood and the blood just moves right up into the tube. And that's because of all the water that's in blood. And so it can actually move up a tube by capillary action. So water has these polar covalent bonds, which allows them to form hydrogen bonds. And that allows water molecules to be sticky, so they can stick to each other, cohesion, or they can stick to other things, adhesion. We talked a lot about osmosis and diffusion. Diffusion is the general property where atoms or molecules will diffuse through a particular area, right? They'll diffuse from an area of high concentration to low concentration, trying to reach equilibrium, where the concentration is equal all over. And that happens with everything. Molecules and atoms are always in motion. It's just some of them move very slowly, others more quickly. But all molecules want to try to diffuse from an area of high concentration to low concentration. Water is a special case of diffusion, involving the movement of water across a semi-permeable membrane, like the cell membrane. So if you have solutes, it dissolved into water, so water is the solvent, and whatever is dissolved in it, like sodium chloride or sugars, is the solute. The problem with many solutes is they're either too big or too charged to get across the plasma membrane. So they need help, like transport proteins. But if you have a case where you just have a membrane, no transport proteins, and water can move through it, but solutes cannot, then water will move. And the water is trying to move to create an equal concentration of that solute on both sides of the membrane. So a hypertonic solution has a high concentration of solutes relative to the solution on the other side of a membrane. A hypotonic solution has a low concentration of solutes relative to the other side of the membrane. And in an isotonic solution, the concentration of solute is the same on both sides of the membrane. So where does the water go? Remember, we learned that water always moves into the hypertonic solution. So water moves into the high solute concentration. So wherever the concentration of solutes is greater, the water moves into the solutes and is trying to dilute the solutes to reach an equal concentration of solutes on both sides of the membrane. In the case of a hypotonic solution, water will leave a cell. If you put a cell in a hypotonic solution, right, the water leaves the cell. So I put a cell like blood cells in pure water. Water will move into the high solute concentration always, right? So in the case of red blood cells, it's a higher concentration inside the red blood cell than outside. So water will move into the red blood cell and cause it to explode. If you put a red blood cell into a salt solution, into a hypertonic solution, water will leave the cell and the cell will shrivel. So water always moves into the high solute concentration. So it moves from hypotonic to hypertonic. And if the concentration is equal on both sides of a membrane, there's no net movement of water. So pH is a measure of the concentration of H plus. The more H plus you have, the more acidic a solution is, and the lower the pH is. So remember, for acids, our pH is less than 7, and bases, our pH is greater than 7. And 7 is neutral, right? Where the concentration of H plus and OH minus is the same. So acids donate hydrogens, and bases will donate OH, or they'll accept the H plus. And so cells have to maintain a constant pH, usually right around pH 7. Remember, each number of the pH scale is 10 times different. So if we go from pH 3 to pH 2, you have 10 times more H plus. It's 10 times more acidic. If you go from pH 9 to pH 10, you're 10 times more OH minus. So that's a huge difference, 10 times. So cells maintain a narrow pH right around 7. And the way they accomplish this is by using buffers, like carbonate buffers or phosphate buffers, which can help maintain a constant pH, either by absorbing excess H plus, right, to prevent, your body or tissue fluid from becoming too acidic, or it can release H plus in order to, bring the pH down if it goes above 7. We talked a lot about the macromolecules, also known as the biomolecules, like carbohydrates, lipids, and proteins. Of the carbohydrates, the simplest carbohydrates are the monosaccharides, glucose, fructose, and galactose. If you join two monos together, you get a disaccharide. Lactose is glucose plus galactose. Maltose is glucose plus glucose. And sucrose is glucose plus fructose. So those are the major disaccharides. And then our polysaccharides are all long chains of glucose molecules. And the three major polysaccharides were starch, cellulose, and what's the one that's in muscles? What's the form of animal starch we find in muscles? Glycogen, right? Glycogen. So animals have the enzymes like amylase, which can break down starch and glycogen, but not cellulose, right? We do not have the enzyme cellulase, which is needed to break down cellulose because the chemical bond is different in the cellulose. So even though cellulose is all glucose, animals cannot get to it, at least not directly. Animals, the herbivores that live on just eating plant matter, have a symbiotic bacteria in their gut, which produce the cellulase to break down cellulose into simpler sugars. And that's how animals can get energy from eating plant matter. Lipids are the fats, phospholipids, and steroids or sterols. We talked about how a fat can be saturated or unsaturated. A saturated fat, like what we see here in this tail, this fatty acid, all single bonds right between the carbons. and they're holding as much hydrogen as possible. So that's what the saturated means, as much hydrogen. If you introduce a double bond, like we see here, well, that reduces the amount of hydrogen, and so it's unsaturated. So, and it also creates a little bend, a kink in that fatty acid. So oils are liquid at room temperature because they're unsaturated, right? They have some of these double bonds, and that creates the kinks, which prevents the fat molecules, the triglycerides here, from packing tightly together. If all the fatty acids are saturated, that means the molecules can come close together and pack more tightly together. And so we have a saturated fat, which is typically solid at room temperature. And we talked about how unsaturated fats are healthier than saturated fats. When it comes to a phospholipid, right, your cell membranes, all membranes are made of phospholipids. They have a characteristic structure where the head, where the phosphate group and the choline, they have charges associated with them. So they're hydrophilic. They love to interact with water. But the fatty acid tails are all hydrocarbon. They're all nonpolar. So they have no charges associated with them. So they hate water. They're hydrophobic because water has all those polar molecules, all those partial negative and positive charges. So a saturated fat or a fatty acid is not going to like to interact with water. So when you mix phospholipids together, you always get a bilayer configuration. Remember we talked about the bilayer. Where the hydrophilic heads face the aqueous environment, the watery environment, and the hydrophobic tails face each other. And that creates a lipid bilayer. Small molecules, nonpolar molecules can diffuse through the bilayer, but things that are big or charged need transport proteins in order to get through. We talked about active versus passive transport. Active transport requires ATP to make it happen. So usually a cell is trying to concentrate something against its gradient. So from low to high concentration. Everything else with passive transport is diffusing from high concentration to low. So they're following their concentration gradient. When it comes to proteins, proteins are made up of amino acids. There are 4 levels of protein structure. Just the sequence of amino acids is the primary structure. When they fold and form alpha helices and beta sheets, those are secondary structures. When everything folds on itself to form a 3-dimensional globular structure, that's the tertiary structure. And for some proteins, that's it. They're done. They're finished. But other proteins require multiple subunits to associate. And that's the quaternary level of protein structure. And we know that we can denature proteins with heat. Right? We use heat. It causes the proteins to unfold, right, to denature, and usually they are destroyed when that happens. We can also use a lot of acid or a lot of base. So a lot of H plus, a lot of OH minus, that will also denature and destroy the protein. Remember, different proteins have optimal temperatures and optimal pHs where they function the best. Many reactions required by living cells either would not occur fast enough for life to exist or they would not occur at all. And that's because energy is required in order for these reactions to occur. We call that amount of energy the activation energy, or the energy of activation. And so lower the amount of activation energy. So they reduce the amount of energy needed for reactants to form products. So that speeds up the rates of reaction. and can speed up that reaction rate on the order of thousands of molecules per second. Now, enzymes are made of protein. Sometimes they also have cofactors and coenzymes. So the protein portion is the apoenzyme, and when that apoenzyme has its cofactor or coenzyme, it's now complete. And we call that the holoenzyme, H-O-L-O, the holoenzyme, because it's whole. Cofactors are typically inorganic elements like manganese and zinc, and coenzymes are organic, and they're usually derived from the B vitamins. But an apoenzyme must have its cofactor or coenzyme in order to be functional. We know that enzymes have an active site, which is where the substrate binds, so the reactions can occur. And enzymes are substrate specific. Only one type of substrate can fit into that active site. So for every biochemical reaction in a cell, you need an enzyme which can make that occur. Feedback inhibition is a property whereby the end product can feed back to an allosteric enzyme, bind to the allosteric site, not the active site, but the allosteric site, change the shape of that enzyme, which changes the shape of the active site, and that enzyme is turned off. And so cells will often do this if they have made too much of a particular product. And the cell does not want to keep making that particular product because that would waste energy. So feedback inhibition is a mechanism to regulate the production of product and save energy. But the binding of the product to the allosteric site is concentration dependent and reversible. So as the cell uses up that product, it no longer binds to the active site, so the enzyme goes back to its normal shape and is able to function again. And that metabolic pathway gets turned back on. So the cell can start making more product. Well, we spent 2 lectures on metabolism, so I'm just giving a brief review here. Same thing with photosynthesis. But metabolism is the sum, a total of all the chemical reactions inside of a cell. And we can put those chemical reactions into 2 groups: catabolic reactions, or catabolism, and anabolic reactions, or anabolism. Catabolic reactions are exergonic, so they're spontaneous reactions. What we're talking about is using some type of food like glucose or another carbohydrate or protein, even fats, break them down, capture the electrons, and use the electrons to make ATP. And in the process, heat also gets released. Anabolic reactions are biosynthetic reactions. The cell is building macromolecules. That requires energy. That energy is ATP. So the catabolic reactions produce the ATP used by cells for biosynthesis, for anabolic reactions. Cell respiration is an exergonic process. It's exergonic because ATP, the hydrolysis of ATP, is used to start the process, right, to make it all happen. So in glycolysis, 2 molecules of ATP are used to start the process. By the end, technically 4 molecules of ATP are produced, but since the cell had to use 2 molecules to start the process, we always refer to the net production of just 2 molecules of ATP. The cell has also made 2 molecules of NADH. The NADHs are carrying electrons to the electron transport chain to make ATP. So our 6-carbon glucose is split into 2 3-carbon pyruvic acids, or pyruvate molecules. Those pyruvates must be decarboxylated and turned into acetyl coenzyme A, or acetyl-CoA. Some CO2 comes off here, right? One of the carbons gets lost from our 6-carbon glucose. Some ATP gets made. Some NADH will get made, and the NADHs are carrying electrons, the electron transport chain, to make ATP. So that intermediate step, the decarboxylation step, only produces NADH. But the electrons in NADH are going to be used to make ATP later. Now the acetyl-CoA is ready to enter into the Krebs cycle, or the citric acid cycle. In a eukaryotic cell, we're inside the mitochondrion. In a prokaryotic cell, we're in the cytoplasm. The acetyl-CoA is going to cycle through a bunch of different intermediate molecules. In the process, each remaining carbon is going to be ripped off one by one and released as carbon dioxide. And the cell is going to make 2 molecules of ATP in the Krebs cycle. And so we said that 2 molecules in Krebs and 2 molecules in glycolysis, 4 molecules total, is not much. Not for all the cellular effort, for all these different reactions, all the enzymes used to make these things happen. 4 ATP is pretty pitiful. But in the Krebs cycle, 6 molecules of NADH are made and 2 molecules of FADH2. So the cell has made 10 NADHs, 2 FADH2, so that's 12 electron carriers. And those electron carriers are going to carry those electrons to the electron transport chain to make ATP. So the cell has ripped apart that original glucose carbon by carbon, captured the electrons, and is going to use the electrons in the electron transport chain to pump hydrogens across the membrane where they accumulate in a high concentration. Then those hydrogens are going to flow back through ATP synthase. And the physical movement of those hydrogens through the ATP synthase is an alternate form of energy that ATP synthase uses to create ATP. So the original chemical potential energy in the covalent bonds of glucose is ultimately captured, right? Those electrons are captured by the NADH and FADH2, and the electrons are used in an electron transport chain to pump hydrogens. So as the electrons are passed from one protein complex to the next in the chain, those electrons, the energy of those electrons, is used to pump hydrogens across the inner mitochondrial membrane of a eukaryote or the plasma membrane of a prokaryote. But the hydrogens accumulate in a high concentration. And we know that molecules want to diffuse to an area of low concentration. So the only way to get back through the inner mitochondrial membrane of a eukaryote or the plasma membrane of a prokaryote is a special transport protein, which in this case is ATP synthase. So as the hydrogens flow back through ATP synthase, that movement of hydrogens is a form of energy that ATP synthase uses to make ATP. And so the majority of ATP made in a cell is made here by electron transport and chemiosmosis. So chemiosmosis refers to the hydrogen ion gradient. And remember, we said the fancy word for that was the proton motive force, because hydrogens are basically just protons, right? The H plus, basically just protons. And the whole process is aerobic because oxygen is there at the end of the electron transport chain to pick up those electrons and combine with some hydrogens to make water. So if oxygen is not present, the electrons stop flowing. The electron transport chain just gets backed up like a traffic jam. It's full of electrons. No more hydrogens get pumped. Even the Krebs cycle mostly stops. So in the case of complex multicellular organisms, that means death. If you're a single-cell microorganism like bacteria or yeast, you can switch to an anaerobic form of respiration called fermentation. And fermentation relies on glycolysis only. And so those two pyruvates will be used to make the end product. If it's a lactic acid fermentation, the end product is lactic acid. If it's alcohol fermentation, the end product is ethyl alcohol. And that's because NADH is still produced in glycolysis, but the electrons in the NADH are going to be used to reduce and produce the end product of fermentation. And so single-cell microorganisms can survive on that. If there's no oxygen, most microorganisms are okay, right? They don't really need it. They have alternate forms of respiration like fermentation. But complex multicellular organisms, even though our cells can switch to fermentation, they can't make enough ATP to survive. And so that's why we will die without sufficient oxygen in around 5 minutes. Our brains die because they need a lot of oxygen. So the ATP produced in glycolysis in Krebs is the result of substrate-level phosphorylation, where an organic compound with a phosphate group transfers that phosphate to ADP to make ATP. And we just said very little ATP is made this way, only about 4 ATPs per glucose, right, just by substrate-level phosphorylation. But all those electrons being used to pump those hydrogens, and then they flow back through the ATP synthase, that's called oxidative phosphorylation. And the cell makes the overwhelming majority of ATP here, usually 32 to 34 molecules of ATP for a maximum of 36 to 38 molecules of ATP per glucose. But remember, that's a theoretical yield, maximum theoretical yield. In reality, cells are not perfect systems. There is some waste. And so the actual amount of ATP is somewhat lower, maybe 28, 29, 30 molecules of ATP total. But don't worry about that. Just know the maximum theoretical yields. We also talked about how NADH, those electrons can produce 3 molecules of ATP, and the FADH2 only produces 2 molecules of ATP, and that was because the NADH donates its electrons at the very beginning of the transport chain, which results in the maximum number of hydrogens being pumped. And it's the hydrogens which are the energy source for ATP synthase. So more hydrogens, more ATP. FADH2 donated its electrons later in the transport chain, so not as many hydrogens get pumped. And so fewer molecules of ATP were produced. So just summarizing, in a eukaryotic cell, glycolysis occurs in the cytoplasm or the cytosol, where glucose is split into pyruvate, also known as pyruvic acid. Just 2 molecules of ATP are made, right, by substrate-level phosphorylation. Our pyruvates are decarboxylated to form acetyl coenzyme A. A little bit of NADH is made, 2 molecules. No more ATP yet, but some CO2 gets released. Inside the mitochondrion, we have the citric acid cycle, or the Krebs cycle, where the acetyl coenzyme A, it cycles through a bunch of different intermediates in the process. 6 molecules of ATP are made, or I'm sorry, 2 molecules of ATP and 6 NADHs are made, and 2 FADH2s. So only a total of 4 ATPs are made, right, by substrate-level phosphorylation. But a lot of electron carriers, right, 10 NADHs, 2 FADH2s, and those electrons will be used in the process of electron transport and chemiosmosis to pump hydrogens across the membrane, where they accumulate in a high concentration and flow back through the ATP synthase. And so ATP gets made by oxidative phosphorylation, and the overwhelming majority is made this way. In a prokaryote, no mitochondria, and so the electrons are used to pump hydrogens just outside the plasma membrane, and they flow back into the cell through the ATP synthase. So here we see an electron transport chain in the inner mitochondrial membrane. We have different complexes in the membrane. As the electrons are passed from complex to complex, right, the electrons have a high energy state to start with. As they move from complex to complex, the energy state drops down. That energy that's released is harvested to pump the hydrogens, and they accumulate in a high concentration, flow back through the ATP synthase, and that allows ATP synthase to make ATP. We said that when the electrons reach the end of the transport chain, there needs to be something to pick them up. That something is oxygen, which combines with hydrogens to make water. Without the oxygen, electrons stop flowing. No more hydrogens get pumped, and complex multicellular organisms die. Single cell microorganisms, no problem, they can switch to anaerobic respiration like fermentation, and they'll survive. And so remember, we learned the overall simplified equation, C6H12O6 plus 6O2 yields 6CO2 plus 6 water plus energy, right? ATP and heat. So that heat helps to increase the entropy of the universe. It also can be used by certain animals to help stay warm and maintain that internal, constant internal body temperature. But this is the overall simplified equation, right, for aerobic cell respiration. So the glucose is oxidized into carbon dioxide. The oxygen is reduced to water. Photosynthesis is another way to phosphorylate to make ATP. It's performed by plants and certain other organisms which contain chloroplasts. But the idea here is that light energy is going to be used ultimately to help generate ATP and NADPH in this case. But the ATP and NADPH are going to be used to make sugar. So the equation for photosynthesis was basically the reverse of aerobic cell respiration. So light energy is needed. 6 carbon dioxides, 6 water, yields C6H12O6, plus oxygen. So the oxygen is actually a byproduct of photosynthesis. So remember, the idea is that light energy is going to be used to excite electrons, to boost them up to a high energy state. And those high energy electrons are going to move through an electron transport chain, right, just like we saw in the mitochondrion, but here we're in a chloroplast. As those electrons move from complex to complex, their energy gets released and harvested to pump hydrogens. Once again, they accumulate in a high concentration and flow back through ATP synthase to make ATP. So same idea, except that light energy is used to excite those electrons. Now the electrons are in these photosystems, in these reaction centers, which contain chlorophyll. And so when those electrons get excited to a higher energy state, they leave that reaction center. So that reaction center is missing electrons. Those electrons have to be replaced, and they come from water. So water gets split, and we call that photolysis or photolysis. And so water gets split to replace the electrons that were boosted and left those photosystems. And so oxygen gets released when the water gets split. And so water is the ultimate source of electrons for photosynthesis. So when those electrons reach the end of the electron transport chain, they are used to make NADPH. So when they reach the end of the electron transport chain, they go to another photosystem, another reaction center, where light energy will again excite them, boost them up to a higher energy state, so they can be used to make NADPH. And the NADPH, along with ATP, will be used in the Calvin cycle to make sugar using carbon dioxide. So a quick overall review of photosynthesis, but the key difference is water is the ultimate source of electrons in photosynthesis. Light energy excites those electrons so they can be passed through an electron transport chain. So the hydrogens get pumped and flow back through ATP synthase to make ATP. When the electrons get to the end of the transport chain, they end up in another photosystem, another reaction center, where again, light energy is going to excite them up to a high energy state so they can ultimately be used to make NADPH. And NADPH is carrying electrons, but it carries electrons to the Calvin cycle, where those electrons can be used along with carbon dioxide to make sugar, right? To make sucrose, starch, or cellulose in the plant. And don't worry about the fine details of the Calvin cycle. Cell cycle and mitosis. Yeah, the conversion messed this up, but well, we know a lot about mitosis because we've learned about it twice in lecture and lab. So some nice review for you guys who are taking the lab exam. So the cell cycle interphase is the largest part of the cell cycle. It's composed of two gap stages, G1 and G2, and a synthesis stage. In the G1 and G2 gap stages, cells are doing their normal cellular activities. In the S phase, the synthesis parts, all the chromosomes are replicated. They're all duplicated in preparation for mitosis and cytokinesis. So mitosis is a nuclear division where two new nuclei are produced. Cytokinesis will divide the cytoplasm and separate the nuclei for two daughter cells. There are 4 or 5 main phases, right? PMAT, prophase, metaphase, anaphase, telophase, or PPMAT, prophase, prometaphase, metaphase, anaphase, telophase. You should be familiar with the key steps or the key events that are occurring. The chromosomes condense in prophase, the nuclear membrane begins to break down. The centrosomes, which contain the centrioles, start to migrate towards opposite sides of the cell. And then microtubules will grow out of the centrioles in late prophase or prometaphase of the nuclear membrane is completely broken down, and other microtubules can attach to the centromeres of those replicated chromosomes. Remember, the centromere is that narrow waist on a chromosome. And also remember that one replicated chromosome is made of 2 sister chromatids. So the microtubules attach to the centromeres. A little molecular motor is formed called the kinetochore, which increases or decreases the length of the microtubules. So the microtubules push and pull on those replicated chromosomes to line them up at metaphase. Then the microtubules pull and separate the sister chromatids, which migrate towards opposite ends of the cells. We call that anaphase. When they reach the ends of the cells, that's telophase. Normally, cytokinesis begins during telophase. In In animal cells, the membrane will pinch in two, and we can see a cleavage furrow, which pinches that membrane. In a plant cell, they have to deposit new cell wall. So we see a cell plate, which eventually will be a fully formed cell wall, which separates those two nuclei. Now, normally cells know when to stop dividing. And here we're focusing just on animal cells. But cells that are touching each other on all sides, we call that density-dependent inhibition or contact inhibition. That tells the cells to stop dividing. And so they form a monolayer of cells, a single layer of cells. They also have anchorage dependence, meaning they need to be attached to a substrate. And if they're not, that tells them to stop dividing as well. And there are certain cells that ignore contact inhibition and anchorage dependence, and the cells keep on growing and dividing and grow on top of each other, forming masses of cells called tumors. A tumor can be benign, which means it's not cancerous, it's not dangerous, or they can be malignant, which means they are cancerous, they are dangerous, they will metastasize and spread and form new tumors. Meiosis is the process that forms the gametes, eggs and sperm. And the main idea behind meiosis is to divide the total number of chromosomes in half. So we're going to go from diploid cells to haploid cells. And this happens after meiosis 1. So meiosis 1, the chromosomes again have to be duplicated. Again, we have sister chromatids, but we have homologous chromosomes now. Half the chromosomes are from the original maternal parent, and the other half of the chromosomes are from the original paternal parent. So we have homologous pairs of replicated chromosomes. They pair up together at synapsis and form tetrads. The adjacent sister chromatids will break and reattach to those switch places. And that's called crossing over. And that occurs in prophase 1 of meiosis 1. Then all the homologous pairs will line up in metaphase 1, and they line up randomly with some of the mommy and daddy chromosomes on each side. And then the homologous pairs separate into two daughter cells. At that point, the total number of chromosomes has been cut in half. We still have replicated chromosomes with sister chromatids, but the total chromosome number has been cut in half. Meiosis 2 is basically like mitosis. We have those replicated chromosomes with sister chromatids, and the cell needs to separate those sister chromatids, just like in mitosis. So all the gametes produced are unique combinations of genes and alleles as a result of crossing over, independent assortment, and segregation. So we get 4 unique gametes in sperm cells. With egg cells, it's just one gamete. Remember, the other 3 polar bodies are just little garbage cells that will be disintegrated. But cells are trying to mix up their alleles, increase variation. So all the gametes are unique combinations of all the alleles in that particular individual. And then random fertilization between 2 unrelated individuals will produce unique offspring. So the whole point of meiosis, increase diversity, mix up the alleles, produce unique gametes. Random fertilization will produce unique offspring. So we're increasing variation, which helps species to adapt and survive. DNA, right, the material which stores all the information needed, right, to form a new organism. We know that we have 4 nucleotides, ATGC, and we know the base pairing is A's with T's and G's with C's, right? So that's the complementary base pairing. A DNA polymerase is the main enzyme, although remember, technically we talked about DNA polymerase 1, 2, and 3, which are bacterial enzymes. Eukaryotic enzymes are more numerous, but the exact same kinds of things happen in the same way. New DNA can only be synthesized in the 5' to 3' direction, so just one direction. About both DNA strands, right? Because remember, DNA is a double-stranded molecule held together with hydrogen bonds, right? Shared between the A's and T's and the G's and C's. And each strand will serve a template for the synthesis of new DNA, of a complementary DNA strand. We talked about how replication is semi-conservative, half old original DNA, half brand new DNA. Replication is a multi-enzyme process. We got helicase, primase, ligase, all required to function properly to replicate DNA. One of the new strands of DNA is synthesized continuously as one long piece, 5' to 3'. That's called the leading strand. But the other strand is synthesized in little pieces called Okazaki fragments, because as we saw in the animation, that other strand of DNA actually has to loop around to come into the proper orientation to allow for synthesis in the 5' to 3' direction. But only little fragments can be synthesized in the 5' to 3' direction. We call those Okazaki fragments. Eventually, they'll all be joined together and sealed with ligase, so they'll be made whole. But during the initial synthesis, one strand is synthesized continuously as one long piece, and the other strand is synthesized discontinuously in a bunch of little pieces, which will all be joined together. Now, all the little pieces actually have a lot of RNA in them because the primers have to be created, and those primers are made of RNA. But remember that the primers, in this case here for DNA replication, create that double-strandedness, which is what DNA polymerase needs to hop on and start replicating to make DNA. Ultimately, the RNA primers will be cut out and replaced with DNA. As I said, all the Okazaki fragments will be made whole, right? They'll all be joined together. But there is RNA in DNA during the initial synthesis because RNA primers are used to give DNA polymerase a place to hop on and start replicating. But the primers will be cut out and replaced with DNA later. DNA polymerase, I should say, is not perfect. It does make mistakes. Mutations do happen. We do not want the DNA polymerase to be perfect because we need mutations to occur. And mutation can be harmful, it can be neutral, or it can be beneficial. And so the accumulation of beneficial mutations leads to adaptation and evolution. So we don't want our polymerase enzymes to be perfect. They must make mistakes every once in a while. And that creates variation and enhances adaptation and survival. So I said there were 3 mates that the consequences were beneficial, harmful, or neutral. The main types of mutations are point mutations like a missense mutation or a nonsense mutation. So in a missense mutation, one of the nucleotides is changed. It changes the codon, which codes for a different amino acid. What the consequences are to that final protein, it depends on what the exact change is, what the new amino acid is. Is it very chemically different? Does it alter how the protein folds? Does it change the active site? If it's a critical protein, that organism will likely die. A nonsense mutation is always bad because that codon gets switched to a stop codon. So that one nucleotide change results in a stop codon. So the protein's going to be short. It's not going to be functional. If it's a critical protein, that organism dies. A frame shift mutation occurs where one nucleotide is either added or deleted, and it changes all the codons after the addition or deletion. So all the groups of 3 nucleotides are all changed, all the amino acids are changed, so the entire protein is changed, or at least after the frameshift mutation. So that's going to be very bad. When it comes to chromosome number, in humans, we have 46 total, or 23 pairs. Remember, 1 through 22 are the autosomes, and 23 is the X and Y, which are the sex chromosomes. All species do not have 46 chromosomes. About a half dozen or so do, at least the ones we know of. Everything else has a different number, and there is absolutely no relationship between chromosome number and complexity of the organism. Now, the tips or the ends of the chromosomes, because remember, eukaryotes have long linear chromosomes. At the tips, they're called telomeres. And the problem with trying to replicate linear chromosomes is that DNA polymerase cannot faithfully replicate to the very end. And so the tips get shorter and shorter with each cycle of mitosis and cell division. That creates problems when your chromosomes get shorter, because ultimately it results in defective genes. So when you start losing your telomeres, at a certain point, your chromosomes are too short. That leads to cellular aging and cell death. Remember, we talked about there's a maximum number of mitotic events that can happen, and we call that the Hayflick limit. And there's some variable estimates of how many cell divisions can occur before the cells reach what's called senescence, where they basically stop dividing and then die. So that limits the human lifespan to maybe 125 years maximum if everything else is perfect. Prokaryotes don't have telomeres because they have a circular chromosome, so there's no loss. A circle gets faithfully replicated each and every time, but our telomeres don't. Unless you have an enzyme that protects the telomeres like telomerase, which we do, and it functions during fetal development, right? Because that fertilized egg, that single-celled zygote, has to go through countless numbers of cell divisions in order to form a fetus. But at some point, the enzyme telomerase gets turned off. If it gets turned back on, it can lead to cancer, right? Because cancer cells are technically immortal. They'll keep on dividing forever as long as they have a favorable environment, because our telomeres don't get short. They are repaired by the enzyme telomerase. So that's why cancer cells are so dangerous, because they just keep on dividing and dividing and dividing. Transcription and translation. Remember the central dogma that DNA is transcribed to RNA, which is translated to protein. The big 3 RNAs are messenger RNA, ribosomal RNA, and transfer RNA. So RNA polymerase is the enzyme involved in transcription. It will transcribe RNA in the 5' to 3' direction. The RNA polymerase knows where to start because there's a promoter sequence and a terminator sequence, which tells it where to stop. In the case of RNA, we have A's, U's, G's, and C's, right? So no T, Wherever you would normally want to put a T, it's a U. Introns are mostly non-coding regions. In eukaryotic genes, exons contain all the coding information. Introns must be spliced out of a messenger RNA using the spliceosome. Sometimes the introns are spliced out differently, alternative mRNA splicing, to produce different messenger RNA transcripts, which are translated to make different polypeptides or different proteins. Messenger RNA in eukaryotes has a 5' cap, usually guanine, and a poly A tail, a bunch of adenines. Those help to stabilize the molecule and allow it to be transported from the nucleus to the cytoplasm. Prokaryotes don't do this because they don't need to. Remember, in prokaryotes, transcription and translation can occur simultaneously because everything's happening in the cytoplasm. There is no nucleus. So remember the genetic code chart. So if you had something like GAC, you find the G on the left here, go across the top to A, and then come down. We find GAC, and it codes for aspartic acid. Remember that there's more than one codon for most of the amino acids. That allows for some flexibility. Like in the case of leucine, as long as you have a C and a U, you always get leucine no matter what the third nucleotide is. We call that the wobble. The code is redundant because of that, because there's more than one codon for most amino acids. There's only one start codon, AUG, which also codes for methionine. There are 3 stop codons. And the start and stop codons tell a ribosome where to start translating and where to stop translating. We talked about autosomal genes, 1 through 22. So most traits are autosomal, right? Because they're on chromosomes 1 through 22. A few characteristics are X-linked, so they're on the X chromosome. Males suffer more because males are XY. So that means they're hemizygous. So I'm not going to go through any of the next problems now because we don't have time, but review the next problem handout. And remember our FOIL technique. Right, for all the possible gametes. And the reason why we get those gametes is because of the law of independent assortment, which happens during metaphase 1 of meiosis 1, where the homologous chromosomes from the original maternal and paternal parents randomly line up at metaphase. And remember, each allele comes from one of your original parents. So like big A, little a. The big A came from, let's say, mom, and the little a came from dad. So they're on separate chromosomes. Homologous chromosomes, the same one, but one that you got from your mother and the other you got from your father. And because they randomly line up at metaphase 1 of meiosis 1, they get sorted independently into the daughter cells. And that's why we get these unique combinations. The mechanism of gene regulation in prokaryotes and a few examples in eukaryotes is the operon. An inducible operon is normally turned off, must be turned on when necessary. A repressible operon is normally turned on, must be turned off when necessary. Inducible operons are for catabolic pathways. Repressible operons are for anabolic pathways for biosynthesis. So inducible operons for those catabolic pathways. If an organism runs out of glucose, it has to find another energy source. We talked about lactose and the lactose operon. So when all the glucose is gone, an alarm signal, cyclic AMP, goes off inside the cell, and the cell starts looking for something to eat. It senses lactose is there. Lactose can bind to the repressor protein, which is bound to the operator, and remove it from the operator. So normally, a repressor protein is bound to that operator sequence. So remember, in between the promoter and the structural genes is that operator sequence where the repressor protein binds. Lactose will bind to the repressor and remove it from the operator. Now RNA polymerase can hop on and start transcribing the 3 genes needed for lactose metabolism: beta-galactosidase, permease, and transacetylase. But the cell will only induce an operon, only turn it on when absolutely necessary, because the genes for glucose metabolism are constitutive. They're always on, always being transcribed and translated. So a cell must run out of glucose and need to use something else for energy in order to switch on an operon, because that takes a lot of energy to synthesize all those new enzymes. So it's not going to happen unless absolutely necessary. In the case of a repressible operon, we talked about the tryptophan operon, a biosynthetic pathway to make tryptophan, an amino acid, but sometimes the cell makes too much. In this case, the tryptophan acts as a co-repressor, binding to the repressor protein. Now the repressor protein can bind to the operator and turn the operon off. But the binding of tryptophan is concentration dependent and reversible. As the cell uses up the tryptophan, it will come off the repressor comes off the operator, RNA polymerase can hop on and start to transcribe, so the cell can make the enzymes in the metabolic pathway that produces tryptophan. By turning off a gene for a biosynthetic pathway, this saves a lot more energy than feedback inhibition. All feedback inhibition does is turn off an enzyme, shut the pathway down. In the case of a repressible operon, when it's repressed, the enzymes are not even being made in the first place, and that takes a lot more ATP. And here's just showing you a lactose operon, right? We have a promoter, the operator sequence, our structural genes for all the different enzymes, a repressor protein normally bound to the operator. Lactose can bind to the repressor and change its shape. So now the repressor protein can no longer bind to the operator, so the RNA polymerase can hop on and start transcribing. But that's only if all the glucose is gone and lactose is present. In the case of the tryptophan operon, it's normally on. In order to get turned off, tryptophan has to bind to the repressor protein. Now the repressor can bind to the operator and block it. But as the cell consumes the tryptophan, it comes off the repressor protein, so the repressor can no longer bind to the operator. RNA polymerase can then hop on and start transcribing and make the genes needed for tryptophan synthesis. So that's how all prokaryotes regulate gene expression. Eukaryotes, a few examples of using operons, but mostly they have many different mechanisms. A lot of them are epigenetic, like histone acetylation, DNA methylation, right, to inactivate genes, or make the chromatin so tight that RNA polymerase cannot get in to transcribe. So that's all the time we have. So that's the whole course in one brief review.